What is the function of the tapetum? Tapetum provides nourishment to the developing pollen grains, or pollen grains are also called as microspores. The male gamete is something related to the pollen grains which are produced by the anther, and the female gamete is nothing but the ovule. Now when the ovule fuses with the pollen grain, we call it as the fertilization. What is the size of the pollen grain? The size of the pollen grain is about 50 micrometer in diameter. They are circular in nature, so that is why we measure them in diameter. Hello everyone, a warm welcome to this concept class session for class 12 biology. I am Dr. Divya, biology faculty. So in this session, we'll learn about few of the topics under sexual reproduction in flowering plants, that is chapter 1. Let's start with the introduction to the chapter. So in flowering plants, we know that they exhibit sexual reproduction wherein the male gamete will fuse with the female gamete to produce the seeds, right? And those seeds are the ones which we sow so that the seeds will germinate and a new plantlet will come out of it. So how does it take place is what we will learn in this particular chapter. So to know that, we need to know about the structure of the male and the female reproductive parts. So this entire chapter depends on that. First, we'll start with learning the parts of a flower. So knowing the parts of the flower is very, very important because the name of these parts will keep repeating in this entire chapter. So that is why. So first, talking about the base of the flower. There should be a stalk which holds the upper parts of the flower, right? So that stalk is nothing but called as pedicel. And right above the pedicel, they have small leaf-like structures which are petal-like structures which are not colored and they are green in color. So those structures are called as the sepals. So one more term for sepals is, it is also called as calyx. So whenever the word calyx comes, don't get confused. It is nothing but the sepals. So right above the calyx, there is presence of a stage-like structures which holds all other parts of the flower. That is, which holds the whorls of the flower. So that is called as the receptacle. So this stage-like structure, we call it as the receptacle, or it is also called as, one more name for that is thalamus. Thalamus. Moving further, the receptacle is the one that acts like a stage or a bed on which all the other four whorls of the flower lies. So talking about the whorls of the flower, the outermost whorl is the sepals or the calyx, which I have already marked. Next to the sepal or the calyx is the brightly colored whorl, which is called as the petals. And the one more name for petals is, it is also called as corolla. It's also called as corolla. So next, inside the corolla, we can find the male reproductive part of the flower and it is called as the stamens. Now, what is the stamen made up of and all that we'll study in the coming slides, okay? So, the male reproductive part is called as the stamen, which forms the andrecium. Andro meaning man, something related to male reproductive part. So, andrecium, that is the male reproductive part, and what it consists of? It consists of stamens. Next, innermost whorl is the gynoecium. So, gynoecium, remember gynecology. Gynecology is something related to the female, right? So, therefore, gynoecium meaning the female reproductive part. So, this is the female reproductive part and it is nothing but called the gynoecium, or we can call it as the carpel. So, or we can call it as gynoecium. Now, what is there in the gynoecium also we'll study in the coming slides. So, as I told you, the stamen is nothing but, this is called as the andrecium. That is the male reproductive part. So, now we know in the gynoecium again, it has the ovary. The ovary has a long style that holds the stigma. Then inside the ovary, ovules will be present. All that we'll study in the coming sessions. Now, important parts of the flower we have marked here. So, moving further, let's start studying about the structures in detail. That is the pre-fertilization structure. So, pre-fertilization. So, what do you mean by fertilization? Fertilization is nothing but the fusion of the male and the female gamete. That is, here the male gamete is something related to the pollen grains which are produced by the anther, and the female gamete is nothing but the ovule. Now when the ovule fuses with the pollen grain, we call it as the fertilization. So now we need to know about the pre-fertilization events. So in pre-fertilization events, we need to know about the male reproductive structure and the female reproductive structure. That is, these are the structures which actually participate in the reproduction or which participate in the fertilization process. So first we will study, in this session we will concentrate on the male reproductive structures. So talking about the male reproductive structure, it consists of the stamens, then comes the microsporangium, and then comes the pollen grain. So each of these we will study one by one. So this is a stamen. So stamen, together we call it as the stamen. Moving further, we'll study about, and can you see that powder-like structure that is there on that? It is nothing but the pollen grains. All these we'll study in the coming slides in detail. Moving further, let's talk about the stamen. So I told you what is stamen. It is nothing but the andrecium. Andrecium means the male reproductive part. So male is represented by this symbol. So male reproductive part of the flower, what is it? It is nothing but stamen. Now what is the stamen made up of? Stamen is made up of anther and it is made up of filament. So filament is the one that holds it. So it is anther and filament, or we can call it as the stalk. It consists of a slender stalk. So what this one? This is the anther and this is the filament. So together this will be the stamen. It's called as the stamen. Now if you look at the anther is a bilobed structure. What do you call by a lobe? Something that looks like this, no, loop-like structure, we call it as the lobe. Okay, this is the first lobe, this is the second lobe. So bilobe means two lobes will be there. So therefore here anther, it is a bilobed structure, meaning it is made up of two lobes. So that is what I have written here. Stamens consists of two parts, that is the filament and the anther. Filament is long and a slender stalk, whereas anther is present at the tip of the filament, and it's a bilobed structure and it is adhered or it is connected to the thalamus. Now you know what the thalamus is, right? I have told you in the previous slide, or the petal of the flower. Sometimes the anthers are connected to the petal of the flower. Now you have studied this in your first PUC. I hope you remember. Let's brush up. So what do you mean when the anther or the filament of the anther is adhered to the petal of the flower? It's called epipetalis. So epipetalis, meaning they are adhered to the petal. So it is epipetalis. And the proximal end of the filament is attached to the number and length of the stamen, and they vary in different species. They can be the size of each of the anthers. So if the anthers are in group, that is we had studied again first PUC, you have to brush up. What is that? Monadelphous, diadelphous, right? Polyadelphous. Now there are groups of anthers and each group will have a different length in the filament. That is the height of the stamen, therefore will vary. So it is like that. And the anther is bilobed. As I've already told you, it is bilobe, made up of two lobes. Now inside, now we know outside the anther what all is there, we know. Okay. So moving further, now what is there inside the anther? So each of the two theca, that is, it is dithecous because each lobe has two theca in it. Therefore, it is called as dithecous. Now, the anther has two lobes, we call it as bilobe. Now, inside the two lobes, there is two thecas. So, that is why 1, 2, 3, 4. So, therefore, it is called as dithecous. Now, each theca is separated by a groove. What do you mean by groove? This is called as the groove. So, something like this, we call it as the groove. So this is called as the groove. Here also these constrictions, so you can put it in that way, the constrictions that are there, they are called as the groove. Now each lobe has its own groove. And the TS, now we take a section of the anther, that is a TS, transverse section of the anther, and it shows that it is tetragonal. Why it is tetragonal? Because it has 4 corners, right? As I told you, 1, 2, 3, 4. So you can see here 1, 2, this is the third one, and fourth one. So therefore it is tetragonous or tetragonal, and in these tetragonal structures there are 4 microsporangia that is located at the corners, and each lobe consists of 1 microsporangia. Now what is in this tetragonal structures? They are nothing but they have microsporangia. So totally how many tetragonal corners are there? 4. So therefore each will have 1 microsporangia. Now how many microsporangias in total? 4 microsporangia. So each will have 1 microsporangia. And what will this microsporangia develop into? So microsporangia is nothing but they are made up of cells. Now some of the cells in the microsporangia, they get developed into pollen sacs, and it is in this pollen sac that the pollen grains will be liberated. Or you can see here in each of the pollen sacs, you can see the pollen grains being produced. Okay. So now we know what is there in the microsporangia, number of lobes, what is there in that, and all that we got to know. Moving further, we will study in detail about microsporangia. This is one of the very important topic, that is about the microsporangia. With a neat label diagram, explain the structure of microsporangia. Always remember, when the word microsporangia comes, now it is something related to the production of microspores. Microspores are nothing but pollen grains in this particular case, in the case of flowering plants. Okay. Now what produces the pollen grain? It is the anther. So therefore you have to draw the diagram of a transverse section of the anther. Okay. Remember like that. So the microsporangia, usually the TS of the microsporangia appears almost circular. Of course, it is lobed, the anther is lobed, but when you see the microsporangia inside, for example, this, they are almost circular, right? They are almost circular. So each of the microsporangia, they appear circular in structure, and it is surrounded by 4 walls. How many walls? 4 wall layers. The outermost is the epidermis, endothelium, middle layer. So we have the outermost layer, it is the epi meaning outer, derma meaning layer or skin. So outermost is the epidermis, then few middle layers they will have, middle layers, and then they will have the endodermis, and then comes the tapetum. Tapetum is actually the nutrient layer that actually provides food to the developing microsporangia. Okay. What is the function of the tapetum? The function of the tapetum is nothing but it produces or it gives nutrition to the developing microsporangia apart from protecting the microsporangia. Okay. So the epidermis, the endothelium and the middle layers, they actually protect the anther altogether, and also they help in the dehiscence of the anther. That is, when the pollen grains, that is some of the microspore mother cells, they produce the pollen sacs. In the pollen sacs, the pollen grains will get produced. Now, when the pollen grains are filled, they just need to burst open and come out. So, for the bursting of the anthers, why the anthers should burst? Because the pollen grains are produced inside the anthers. So, when the anthers have to burst, these 3 wall layers that are there, or the 4 layers that are there, that needs to burst open. So, therefore, all these layers, they help in, especially the epidermis, endothelium and middle layer, they help in dehiscence. The bursting of the anther is called as dehiscence, wherein once the anther bursts, all the pollen grains will just spurt out. So that is it. So therefore, for the release of pollen grains, these 3 layers, that is the epidermis, the middle layers and the endodermis actually helps, or the endothelium actually helps. But tapetum's function is to provide nutrition to the developing pollen grains and the microspores as well. Okay. So this is about the microsporangia. Moving further, I told you the tapetum. What is the function of the tapetum? Tapetum provides nourishment to the developing pollen grains, or pollen grains are also called as microspores. What did I tell you? Pollen grains are also called as microspores. Micro meaning minute. Okay, so spores are the ones which bring about fertilization. So microspores. And where are these? The structures which produces the microspores are called as microsporangia. Therefore, what is the microsporangia? It is nothing but the anther. Okay. So the cells of the tapetum, it consists of dense cytoplasm. So it is filled with thick cytoplasm and it is multinucleate. Multinucleate means it has more than one nucleus. It is multinucleate. So this is the structure of the microsporangium. This is very, very important. Next, we know what is there inside the microsporangia. What is there? Pollen sacs. And in the pollen sac, microspores are there. So we'll talk about the pollen grains. Okay, pollen grains, they are actually the male gametophyte. And male gametophyte gametes are two types of gametes are there. One is the male gametophyte and the female gametophyte. Male gametophyte is nothing but the pollen grain. Here I'm talking about the flowering plant. So therefore male gametophyte will be the pollen grains. Female gametophyte will be the ovules. Okay, now I hope you have understood. But if I talk about human beings, it will be male gametophyte will be the sperms. Female gametophyte will be the egg or the ova. Understood? No, but it is about plants that we are talking in this particular chapter. So what is the size of the pollen grain? The size of the pollen grain is about 50 micrometer in diameter. They are circular in nature. So that is why we measure them in diameter. And the pollen grain is filled with cytoplasm, and in the cytoplasm of the pollen grain, so the pollen grain is filled with cytoplasm and it is covered by a plasma membrane. The cytoplasm is covered by a plasma membrane. And the viability of the pollen grains varies depending on the temperature and humidity. So viability means the living capacity. Viable means they are living. Non-viable means they are dead. So the viability or the living state of the pollen grain can always depend on the temperature and the humidity. And also the viability of the pollen grain depends on the how hard the exine or the outer covering of the pollen grain is. Moving further, the pollen grain. Now we'll talk about the structure of the pollen grain. We got to know that it is 50 micrometer in diameter and all that, and we got to know that inside it is filled with cytoplasm and the cytoplasm is covered by a plasma membrane. All that we got to know. Now, apart from having that, the pollen grains are also covered by two important layers. One is called as the outermost layer, which is the exine, and the inner layer, which is the intine. So this spiny layer that is there, it is the exine, and inner to that it is the intine. Exine is usually hard, it has spicules, it has lots of depositions on it, and even there in exine sporopollenin will be there. So sporopollenin is actually a yellow fluid substance that actually protects the pollen grain and it increases the viability of the pollen grain, and also it makes the pollen grain so tough so that it can be resist again high temperature, high humidity or very less humidity. It can resist again high acidic conditions, any chemicals and all that. So that is why pollen grains can be preserved for many years. So talking about the exine, as I told you, it is the hard outer layer and it is made up of what? It is made up of sporopollenin. This word sporopollenin, no, please remember in this chapter, it is very important. And the sporopollenin, as I told you, it can withstand high temperature, strong acids and alkali. And wherever there is sporopollenin on the exine, their germ pores will be absent. And wherever sporopollenin is absent, then the germ pores will be present. So the apertures or openings that are present on the exine, wherever sporopollenin is not there, that is called as the germ pores. And the exine, it is so very beautiful. It has different patterns. Can you see here? Here itself I have put how beautiful the different patterns of the exine are, right? It is because their spines and their crustaceous layer and all that is there that gives it a different pattern and design. So it's very beautiful to observe the pollen grains under the microscope. Next, moving to the next layer that is present below the exine, it is called as the intine. Intine is not that thick as exine. It is thin and it forms a continuous layer. Why intine is a continuous layer? Because it doesn't have any germ pores or any apertures are not there. Therefore, it is a continuous layer. And what is it made up of? It is made up of cellulose and pectin. So here, exine is made up of or consists of sporopollenin. Intine consists of cellulose and pectin. Exine is not a continuous layer. Intine is a continuous layer. Exine has germ pores or apertures. Intines do not have germ pores or apertures. So I'm just telling about the differences between the two. Okay. Moving further, a mature pollen grain if we take a section of a pollen grain or look into what is there inside the pollen grain, it has two cells. One is a vegetative cell and a generative cell. So you can see here, this is the vegetative cell. The vegetative cell is comparatively larger when we see the generative cell. And it is in this generative cell, actually the two male gametes will be present. So where will the male gametes be present? In the generative cell. Okay. So in the vegetative cell, it will just have the nucleus will be present, and also the vegetative cell, as I told you, it is bigger and it is filled with lot of food reserve, and it has a large irregularly shaped nucleus. So because it is filled with lot of food reserves, the main function of the vegetative cell is to provide nutrition to the male gametes until they travel into and reaches the female gamete. Okay. So generative cells. Generative cell is small and it floats in the cytoplasm of the vegetative cell. Now inside the vegetative cell, what all is there? It has the nucleus and also it has the cytoplasm. Now inside the cytoplasm of the large vegetative cell, you can see the generative cell floating in the cytoplasm of the vegetative cell, and generative cell is smaller when compared to the vegetative cell, and it has a spindle shaped and filled with dense cytoplasm and also a nucleus. So moving further, now we know what is the outer structure of the pollen grain, what is inside the pollen grain, and all that. All this you need to remember because for the next sessions it will be useful. Now we'll talk about the advantages and disadvantages of pollen grains. So pollen grains are rich in nutrients. So therefore they are used in making pollen tablets, and these pollen tablets are just used as food supplements. That is, to get proper nutrition, you can take this pollen tablet. So these are the pollen tablets that are there, which can be just swallowed as a supplement. And pollen consumption, it has also been claimed that it is used by athletes, and also pollen tablets are given to race horses because it has so much of nourishment in them that they give them the extra energy to participate in the race and all that. So these are the uses of pollen grains or advantages of pollen grains. Now pollen grain has its own disadvantage as well. What are those? Let's look into. So pollen grains of many plant species, say for example, we have the carrot grass or the parthenium. So the pollen grains of that is said to cause a lot of respiratory disorders for both animals as well as human beings. So they cause severe allergies, they can cause bronchial pain also, and also they can lead to overexposure to these pollen of some of the plants. It can lead to lot of harmful or chronic respiratory disorders such as asthma, bronchitis, etc. Now, once this happens, there is no cure for that, right? We have to just take medications and keep it under control. I have given here the example, already told it to you. Parthenium, which is also called as carrot grass, it was not there at all in India. But what happened was, during many years ago, during the import of wheat from other country, what happens, along with the wheat, they came as a contaminant. They mixed with the wheat seed and it came into our country. And weeds, you know, wherever you want, you can grow them. So it started to develop in the wheat fields and then it spread throughout the country. Now in India, any wasteland or roadsides, anywhere, or even in farms also, some of the farms, if you see, you can see a lot of parthenium develop. Even in forest area also you can find the parthenium growing. So that is how it has become an alien species, destroying a lot of other plant species and also causing a lot of respiratory disorders to humans and animals. So this is about the parthenium. So parthenium is one of the pollen allergens. It causes pollen allergy. So this was about the section. Next section will continue about the female gametophyte. So far in this session we studied about the male gametophyte. That is, in the male gametophyte we learnt about the structure of the stamen and we learnt about what is there in the stamen. So we learned that it is made up of anther and it is made up of the filament. Now in the anther, microsporangia is there. In the microsporangia, microspores will be developed, which are nothing but the pollen grains. Then we got to know about the structure of the pollen grain. Inside the pollen grain, we got to know that it consists of the vegetative cell and the generative cell, and we got to know that the pollen grain is covered by two important layers. That one is the toughest layer called as the exine, and other one is the thin layer called as the intine. And also we got to know about the advantages and disadvantages of the pollen grain. So in the next session, we'll concentrate completely on the female reproductive structure, wherein we'll study about the pistil or the gynoecium, and we'll study about the megasporangia. There in male reproductive structure, we studied about the microsporangia. Here we'll study about or the pollen grain, right? Here we'll study about the megasporangia or the ovule, and we'll also study about inside the ovule what the female gametophyte that is there, which is called as the embryo sac, that we will study. So see you in the next session with these topics. So I hope you found this session quite useful. So see you in the next session with these two topics under the same chapter. Thank you.